So one of the first questions anybody might ask is, why do we care about cryo-EM? And I like to show this slide, which shows the enormous and breathtaking growth of cryo-EM over the last few years. And that's sort of illustrated. It's not the only measure, but one of the measures is the number of structures in the protein data bank, the number of protein structures that have been docked into that, submitted to the protein data bank. And you see this incredible growth here. This is a logarithmic scale. And cryo-EM has just shot up, and we all know why, but I'll talk a little bit about it just to give some historical background. And it's on a trajectory to catch up with X-ray crystallography over the next 5 years, and we certainly hope it will. So that's one of the reasons we may care about cryo-EM. And another reason, of course, is very well illustrated by the current crisis we're all going through with COVID-19. These are two papers of that dealt with the COVID-19, SARS-CoV-2 spike protein, the protein on the outside of that virus. And those structures came out 3 weeks after the gene was delivered to those groups. So that's incredibly breathtakingly fast. And of course, now there's hundreds and hundreds of more structures. But cryo-EM has had an enormous role to play in both vaccine development and therapy development for COVID-19. So that's yet another reason we would care. And then finally, you've heard both from Giovanna and very much from Annetta in the last webinar, all of the other things that cryo-EM can do. I'm going to mostly focus today on the high resolution structure determination for both proteins and chemical compounds, but I'll come back to this